Hey guys, so I'm going to go over that first section of chapter 4, section 1. It is about microscopy, and I know it's kind of it's tough to look at or to understand on your own, but I want you to see, I want to highlight what the importance of it is and what it's trying to get you to understand. So, as you know, I know that in biology class you've always looked through a microscope at cells, and I know you probably haven't seen very many details when you've looked under the microscope. You've seen something like this when you look at a light microscope. So how in the world do scientists know all the details of the organelles inside of the cell? So remember, when we're looking at microbiology, everything that is being told to you in your textbook and any picture you see, it had to be figured out by somebody. So your question you should be asking is, how did they figure that out? And the first step is through different types of microscopy. So this microscopy that you're used to, what we use in the labs is this light microscope. And what that does is it just puts light through the sample, and you have like kind of magnifying glass, and you're able to see what's there. It's not very detailed. As you can see, this is the limit of the light microscope. So when we're looking at cells, you can't really see the details of inside of the organelle. So if you look at this plant cell, we know that those are chloroplasts because I told you that they're chloroplasts. We know that this is the cell wall because you've been told that's the cell wall, but how do we see details like how we study it in biology class when we see our diagrams? So some other types of microscopy that didn't come around until the 1950s, which seems like a long time ago, but not that long ago in the realm of science. So we have a new technology, and it's the electron microscopy. And what that does is they put a film of metal over the sample, and then they shoot electrons at it. And based off of the kind of like the wavelengths they see with the electrons, it shoots back an image at them, so you can see a topographic kind of map of what is actually there on the organelle inside of the cells. So you get images like this and like this. So as you can see, the detail is a lot different between the light microscopy and the electron microscopy. And with the electron microscopy, if you look here, this is a chloroplast, you can start seeing the inner membranes. Here's the vacuole. So you can start seeing some of these structures that we talk about in our books. Another thing when it comes to really understanding the structure and function of things that are going on inside of a cell, you kind of have to put a lot of different scientific techniques together to come up with an answer. So we can see the chloroplast here, but how do we know what it does? And so we have technology like this fractionation, and what that does is you put some cells into a tube and you put it into a centrifuge, and a centrifuge spins it really, really fast, and so it separates the different parts of the cell based off of their density. So the more heavy pieces are going to go down to the bottom, and the lighter pieces are going to float up at the top. So as you spin it more, you start getting these different layers of organelles within the cell. And then you can take a sample from that. So if I took this sample, so in this example it says that's fragments of the endoplasmic reticulum, you can study that. You can take the biochemistry of it, so you could test and see what substances are in there. So if we see a lot of lipids, then we know that lipids have something to do with the endoplasmic reticulum. And so because of that, taking the biochemistry and these divisions of the cell parts, we can start studying what the function is of these organelles. For example, if you separated a chloroplast, like from this picture, if you took this sample of tissue and you put it into a centrifuge to test it, you would definitely figure out when you looked at the biochemistry that the chlorophyll, that the pigment that's making these plant cells green, would definitely be connected with the layer that has the chloroplast in it. And so you would be able to come to a conclusion that chlorophyll must be in the chloroplasts. Another type of technology that we also use to put all the pieces together, it's like one big puzzle, it's kind of fun, is this. This is called, they use, let me spell it right, it is fluorescence. And we have the technology now. It actually comes from a jellyfish, so jellyfish glow. We can take the protein that glows in the jellyfish and we can put it on a marker in a cell, a piece of a cell. So then when a cell grows and a cell is formed, that fluorescent molecule, that protein, is attached to what we know we attached it to. So when we see it inside of a cell and we know we attached it to, in this case, it's a protein found in Golgi bodies, we know that what we see in this picture glowing green with the fluorescent tag is a Golgi body. And what we see here, what scientists found out by looking at this image, is that the pieces of the Golgi body are glowing, but we also see the cell membrane. We see the outline of the cell membrane. So that leads the scientists to believe that the Golgi has some kind of function with the cell membrane, because we put a tag on the Golgi body, on a protein in a Golgi body, and we are also seeing that that protein is in the cell membrane. So the Golgi body has to produce that protein. It has to be part of producing and forming the cell membrane. So I just want you to keep in mind that when we're going through these cell parts, always think, what made the scientists know that this is true? Give me the evidence. And always think about all these pieces, all these science methods, these scientific methods, whether it's microscopy, whether it's fluorescence or fractionation. All of these put together are clues so that we know when we read about the function and structure of organelles, all those pieces have been put together for you. Alrighty. Thanks, guys.